During the first stage of cellular respiration, which is glycolysis, it can occur with or without oxygen. But when oxygen is present, the pyruvate produced through glycolysis will be oxidized into acetyl-CoA through Link reaction. This is because most of the energy are still remain in pyruvate. So when oxygen is present, the pyruvate in eukaryotic cells enters a mitochondrium where the oxidation of glucose is completed. Pyruvate molecule is a charged molecule, so it will be transported into the mitochondrium through active transport and reach at the matrix of mitochondrium. There are three reactions occurs in Link reaction, which is catalyzed by a multi-enzyme complex. The functional group, which is the carboxyl group of pyruvate, will be removed and released as carbon dioxide. So this is the first step in which carbon dioxide is released during respiration. And the remaining two carbon fragments will be oxidized and the electron is transferred to NAD+ to form NADH, which is a high energy molecule. The last step, coenzyme A, which is a sulfur-containing compound, will attach to the oxidized two carbon fragment to form acetyl-CoA, which is a high energy molecule. This is why the Link reaction is also called as oxidative decarboxylation, because it involves the decarboxylation process and oxidation. Okay, so from one pyruvate, it will release one carbon dioxide from decarboxylation and form one NADH through oxidation, and finally produce one acetyl-CoA, which is two carbon molecule, after the addition of coenzyme A. But for glycolysis process, we always begin with one glucose. So one glucose is split into two pyruvates. So from one glucose, after the Link reaction, it able to produce two CO2, two NADH, and two acetyl-CoA. The Krebs cycle is called as citric acid cycle, which is actually the second stage of cellular respiration, but actually it's the second stage of aerobic respiration also, because it only can occur with the presence of oxygen. Krebs cycle or citric acid cycle is defined as a series of chemical reactions in aerobic respiration, in which acetyl coenzyme A is completely degraded to CO2, water, and ATP. So for one citric acid cycle or for one Krebs cycle, it will produce one ATP, three NADH, one FADH2, and two CO2. But we continue with the process of glycolysis, which begins with one glucose. So it requires two times of citric acid cycle, or involve two molecules of acetyl-CoA. So this is the final result of one glucose. There are total of 8 states in one Krebs cycle or citric acid cycle, and each step is catalyzed by specific enzyme. But in our syllabus, we don't need to remember the specific enzyme in detail. Just need to focus on what type of reaction occurs in each step. Acetyl-CoA is a high energy molecule, which means they carried the coenzyme A that attached to the two carbon fragments with an unstable chemical bond. When this unstable chemical bond is broken down, it will release the energy, and the energy will be used by the two carbon acyl group to combine with the four carbon oxaloacetate and produce a six carbon citric. Citric is the ionic form of citric acid. This is why Krebs cycle is also known as citric acid cycle, because citric is the first stable molecule formed during the citric acid cycle. Isomerization occurs during step 2, where one citrate will undergo removal of one water molecule and followed by addition of another water molecule to form its isomer, which is isocitric, which is also a 6-carbon compound. During step 3, oxidative decarboxylation occurs because the 6-carbon isocitrate will undergo oxidation by transferring the electrons to Na+ and form NADH. The oxidized molecule will undergo CO2 removal and form a 5-carbon compound, which is alpha ketocrutarate. During the next step, oxidative decarboxylation occurs also. Alpha ketocrutarate will undergo removal of one CO2 and form a 4-carbon substrate, and this molecule will undergo oxidation by transferring electrons to Na+ and form NADH. And now the oxidized substrate is attached with one coenzyme A through an unstable chemical bond and form succinyl-CoA. Succinyl-CoA is a 4-carbon molecule that contains high energy. Enzyme A found in the succinyl-CoA is high energy molecule and is displaced by phosphate group. But the chemical bond is unstable and easily broken down, which will release the energy to transfer the phosphate group to GTP and form a GTP. GTP is a high energy molecule which is reactive or unstable. So it will release one of the phosphate group to ATP and produce high energy molecule ATP. So this is a step involve substrate level phosphorylation because they produce the ATP by using the energy found in the substrate succinyl-CoA. So after step 5, succinyl-CoA is converted to succinate. And step number 6, succinate undergo oxidation, or succinate is oxidized by transferring the hydrogens and electrons to FAD and produce another electron carrier or high energy molecule, which is FADH2. So the 4-carbon succinate is oxidized to produce fumarate, and the 4-carbon fumarate will accept one water molecule and undergo rearrangement of bonds to produce malate. Each malate will be oxidized by transferring the electrons to Na+ and produce NADH, and during this step, oxaloacetate is regenerated, and oxaloacetate will enable the repetition of citric acid cycle when the acetyl-CoA enters to the cycle. Since that we study the aerobic respiration that begins with glycolysis, that one glucose able to form two pyruvates, and each pyruvate with the presence of oxygen, they will enter the mitochondria to complete the link reaction and also the Krebs cycle. This is why if we focus on the two pyruvate that will produce the two acetyl-CoA that complete the two cycles of Krebs cycle, it will produce two molecules of ATP through substrate level phosphorylation, and the sixth molecule of carbon dioxide will be released through the decarboxylation that happens in link reaction and also the Krebs cycle, and also the electron carriers, which are NADH, that can be formed during the link reaction, and also three steps in the Krebs cycle, and two molecules of FADH2 that is produced during the Krebs cycle.